Thanks for taking the time to follow along on this recorded webinar entitled Monitoring Cell Health in Real Time. My name's Roy Edward. I'm the Sales and Marketing Director at Biostatus Limited, and this is a repeat of a presentation that was made at the Royal Microscopical Society Advances in Cytometry meeting in November 2015 in London. For those of you not familiar with Biostatus, this slide just gives some brief background information on the company. But to start, I'd like to explain the core features of the product DRAQ7 that lead to its use as a real-time monitor of cell health. Accordingly, by way of background, it may be helpful to understand the parent molecule DRAQ5, which is a cell-permeant DNA binding probe that conveniently fluoresces in the far red, combining easily with commonly used chromophores such as fluorescein, phycoerythrin, Texas red, and rhodamine. It has ideal properties as a reagent, a pure synthetic molecule with remarkably low photobleaching, stable at room temperature and in ambient light, and importantly highly compatible with physiological buffers. DRAQ5 is cell permeant. That is, it's highly lipophilic, yet it is surprisingly soluble in aqueous solution. It has high affinity and specificity for double-stranded DNA, intercating preferentially at AT2 sites, as shown in the figure here. Usefully, it has minimal RNA binding and consequently is not seen and therefore pumped from cells by, for example, ABC transporters, making the nuclear labeling stable and less time-dependent. DRAQ5's core application is the rapid, specific and stoichiometric counterstaining of DNA of living and fixed cells for flow cytometry or fluorescence microscopy. Since its commercial release in 2001, DRAQ5 has been cited in more than 4,900 peer-reviewed papers. Following a number of requests from customers for a viability probe with the spectral and reagent properties of DRAQ5, our R&D team set about developing what became DRAQ7. DRAQ7 retains the DNA binding and spectral properties of the DRAQ5 parent molecule, yet significantly differs from DRAQ5 in that it has been rendered cell membrane impermeant and hydrophilic. In principle, this means that as a far-red DNA binding viability probe, DRAQ7 should elicit minimal phototoxicity, while the parent molecule DRAQ5 can be conveniently used as a positive control without resort to mechanically or chemically induced cell permeability. At this point, a reasonable question would be whether DRAQ7 does the core job of a viability probe that it was designed to do. This slide shows that to be the case. It sensitively reports styrosporine-induced apoptosis in a dose-dependent manner, with clear separation between negative and positive cells. Whilst there is no appreciable non-specific labelling of intact cells of the kind that is often seen with propylium iodide, from data published by Smith and colleagues in 2013. DRAQ7 can be combined with other functional cell health probes, here shown with the mitochondrial membrane potential probe TMRM from Akagi et al., also from 2013. As noted at the bottom of the slide, this required performance of a standard viability probe is achieved in the context of ready-to-use convenience. The previous data shows that indeed DRAQ7 does perform as a viability probe, and leads to the obvious applications including partnering with an Exin5 in place of PI and an iSAGE protocol for the enumeration of CD34 positive, CD45 positive progenitor cells in place of 7AAD. Beyond this standard performance, it was already apparent from internal data that DRAQ7 had no appreciable impact on cell proliferation when included in the medium of long-term cell cultures. In the context of anti-cancer drug cytotoxicity, scientists at Imogen Biotech in Manchester have deployed DRAQ7 in their assay development program for personalised medicine strategies, as shown here. This describes DRAQ7's compatibility across different cell types and its lack of interference with the efficacy of the drugs tested. As already stated, DRAQ7 intercalates with double-stranded DNA, consistent with its parent DRAQ5, and performs equally well as a fixed-cell nuclear counterstain. Therefore, it can be used to measure the failure of cell membranes cell by cell, rather than at bulk level that would be done by measuring released ATP. Moreover, this discrete cell-by-cell measurement and spectral separation allow one to unpick the temporal and spatial sequencing of apoptotic and necrotic events. Looking closer at the spectral properties of DRAQ7, a far-red viability probe is widely compatible with visible range chromophores in both 2D and 3D fluorescence microscopy and in flow cytometry. Helpfully, the red excitation and far-red emission of DRAQ7 limits the degree of scatter and phototoxicity alongside the aforementioned multicolour and cross-platform compatibility. Shortly after DRAQ7's launch, a thorough peer-reviewed investigation of real-time cell health monitoring was undertaken. One key element of this work was the exploitation of a highly sensitive test for DNA-binding molecules developed by Professor Zbigniew Darzenkiewicz. This determined that DRAQ7 was non-toxic to cells over several days of exposure at concentrations well above that of the standard protocol, and importantly showed that DRAQ7 did not passively enter cells. Dose response profiles for anti-cancer agents were unaffected by the presence of DRAQ7 in the cell cultures, yet DRAQ7 remained a sensitive real-time readout of cell damage or cell death. The authors were able to combine DRAQ7 with common fluorescent functional probes such as an Xn5 FITC and TMRM without concerns of spectral overlap, enabling easier assay design. One practical application of DRAQ7 as a spectrally separated viability probe that doesn't interfere with cell function is described here. The background to this study is that the measurement of a small population of cells, the so-called side population of stem cells, is sensitive both to time and to the presence of dead cells. In this context, the properties of DRAQ7 are then ideal to allow a multicolour, temporal study of cellular efflux, of Hoechst dye, and if sorted, allow downstream molecular or cell culture studies. Intact cells do not label non-specifically with DRAQ7, while membrane-compromised positive cells are some 2 orders of magnitude brighter than the intact cells. Subsequently, a number of reports have exemplified the use of DRAQ7 for real-time cell health monitoring. Liang and colleagues from the University of Liverpool followed apoptosis using time-lapse imaging of DRAQ7 and an Xn5 treated cultures for up to 72 hours to better understand the value of USP30 as a target for anti-cancer drug discovery. Ware and colleagues from Houston and Swansea used DRAQ7 to monitor nanoparticle toxicity over 24-hour periods. In the context of high-content screening, DRAQ7 has been demonstrated on the SN Bioscience IncuSite platform to monitor apoptosis and death in real time by 2 laboratories at Mount Sinai School of Medicine by Stratikopoulos and colleagues and Reno and colleagues, both in 2015. In a practical application of a potential therapeutic cell-killing methodology based on intracellular magnetic hyperthermia, Blanco Aduyar and colleagues from University College London used DRAQ7 in real-time cell health studies to elucidate the delay in cell death after treatment, while Spear et al. Demonstrated an ability to induce apoptosis in macrophages infected with Legionella and thereby propose a host-driven means of mitigating the extent of bacterial expansion. DRAQ7 has been demonstrated in real time or long-term study of cellular toxicity for common anti-cancer agents against patient-derived microtissues in a project by Imogen Biotech looking towards personalized medicine. An ex vivo method used by Marciniak et al. Employed DRAQ7 to monitor tissue health in pancreas slices over 7 days. DRAQ7 has been utilized to exclude dead and damaged cells from cell sorting as a preparative technique for molecular analysis, in this case by Preisel et al. For RNA sequencing. Exclusion of fragmented RNA, or in fact RNAs, from such cellular events may be important in reducing background. This also lends weight to DRAQ7 having no interference of the polymerases involved in the amplification procedures. And finally, a recent application of DRAQ7 developed by Wang et al. Was for the detection of subcutaneous extracellular DNA in mice that had been vaccinated using a novel combination of non-ablative, lasing and single-use multi-pin inoculation tool. A further table here shows the variety of cell types to which DRAQ7 has been applied in the early publications, demonstrating its wide utility. In practice, DRAQ7 is very easy to use for long-term cell health applications. As a guide, concentrations between 1 and 1.5 micromolar are appropriate, but of course these should be verified by titration. DRAQ7 has wide compatibility with cytometers, fluorescent microscopes, high-content screening imaging platforms, and the popular Essen Bioscience IncuSight device. As described in the earlier slides, DRAQ7 is optimally red excited and detected in the far red or near infrared regions of the spectrum. In summary, DRAQ7 allows multi-parameter cell health apoptosis assays. DRAQ7 only labels the dead cells, one by one, when compared to other methodologies such as ATP or Alamar Blue. DRAQ7 is compatible with 2D and 3D assay formats, including ex vivo tissues. DRAQ7 uniquely permits real-time long-term analysis due to its ultra-low toxicity properties when compared to conventional alternatives including DAPI, PI, 7AAD. Also, importantly, DRAQ7 has shown no interference or synergism with therapeutic agents used in parallel with it. To find out more about DRAQ7 and related products, please explore our website. And we are always happy to receive your questions and will endeavour to answer these within 24 hours, however complex each of these might be. Thank you for taking the time to learn more about DRAQ7 for real-time cell health assays.